Okay, so welcome to this next video on CFTR, and in this video what we want to discuss is the synthesis of CFTR and how we traffic it to the apical membrane, and particularly what we want to focus in on is how a very common mutation in cystic fibrosis, namely the delta F508 mutation, can disturb this process. Okay, so firstly let's begin with the cystic fibrosis transmembrane regulator gene, which is on chromosome 7 in the nucleus. So if we have some cell here, so here's our epithelial cell, then in the nucleus what we have is we have 23 pairs of chromosomes. Now on chromosome 7 is where you will find the CFTR gene, so let's take chromosome 7. So what we have here is chromosomes have two arms, and one is a short arm, this top one here, so this is the short arm of the chromosome, and the other is the long arm. Now the short arm is referred to as the P arm, and the long arm is referred to as the Q arm. So this is the P arm, and this is the Q arm. Right, and this is chromosome 7 that we're talking about. Now, somewhere in chromosome 7, you will find the CFTR gene. So let's pull it out and imagine we've got it over here. So we've got this strip of DNA here, which is going to represent our CFTR gene. So I'll just complete the double helix. Okay, so let's imagine that, where's my highlighter here? Let's imagine that this represents the whole CFTR gene. So all of this is the CFTR gene. Now the CFTR gene is a very, very big stretch of DNA. It consists of 250,000 base pairs. So basically, if you count the number of organic bases that are paired in this strip of DNA, and you go all the way along from the start of the CFTR gene to the end of the CFTR gene, you will count around 250,000 of them. So that is a big strip of DNA. Now, the protein is not coded for by a gene that size. Most of it is non-coding. So most of it is what are known as introns. So basically, there are strips of this DNA that are actually going to be used to create the CFTR protein, and those are referred to as introns. So let me draw them on. So basically what we're going to have is little strips of this that are actually used for coding and used to actually make the CFTR protein itself, and all the bits in between are just junk basically. They're not going to code for anything. So, these are known as exons. So, between these two lines is an exon, a bit of the DNA which we're actually going to use to construct something. And basically, in the whole CFTR gene, in this whole 250,000 base pairs, there are 24 exons. And all the bits in between, so these big bits here, these are introns, or non-coding DNA. Right, so when we transcribe this huge, great CFTR gene into mRNA, what we're going to do is we're going to transcribe all 250,000 base pairs, and we'll end up with a strip of mRNA like this. And if I mark on the exons on here again, so here are these 6 exons that I've drawn. Of course, in reality, there'll be 24 of them. But I don't want to have to draw 24. So these are the exons. These are the bits we actually want to use to make our CFTR protein. And all the bits in between are introns. So this is an intron, and this is an exon. Right. And this overall is a piece of mRNA, but this is actually what is known as pre-mRNA, because it's the bit that we've just literally transcribed from the DNA. Before we're actually going to make it, before we're actually going to use it to make protein I, before we're actually going to put it through a ribosome and let translation occur, what has to happen is you have to cut out all of these introns, these bits that aren't going to code, and just stick together the exons, basically, to make what is known as a mature piece of mRNA. And that process is known as splicing. So this is the process of splicing. So basically what we're going to do is we're going to take this intron here, the start of this exon, we're going to get rid of the introns. So this is this first bit of DNA, of RNA that we actually want to keep. Then we're going to take the second bit from here. Then we're going to, so I'll just mark it with this. Then we're going to take the third bit from over there. We'll end up with the fourth bit from here, 5th bit on, 6th bit on. So basically we're going to cut out all the clutter and just keep the coding bits and then join them all together. And then what we get is a mature piece of mRNA. This then goes on to the endoplasmic reticulum, which is a membrane-bound structure which is very closely associated with the nucleus of the cell. So this mRNA is going to go up into this endoplasmic reticulum, and it's going to be translated in this endoplasmic reticulum. So I'll draw this piece of mRNA here. So it's just a strip like that. And basically what is going to happen is that you're going to have a ribosome here, which is going to use this mRNA to make you your CFTR protein. So you end up with your CFTR protein. So I'll draw my CFTR protein here, okay? So this is the CFTR protein. Right. And now what I want to discuss is what is going to happen to the CFTR protein next, basically. So next what happens is that in the endoplasmic reticulum, the CFTR has what is known as core sugars added onto it. So basically what is meant by a core sugar is, so if I do an example down here, if you remember from biochemistry what a glucose molecule looks like, it's basically this hexagon molecule. And you'll remember that if you make starch or glycogen out of glucose, what you effectively do is you bind glucose to glucose over and over again. And you bind the hydroxyl group on this first carbon, because this is actually an oxygen over here. So this is counted as the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and the sixth carbon actually sticks off up there. Basically, you bind the hydroxyl group of the first carbon to the hydroxyl group of the fourth carbon, and you create these long repeating polymers, basically, structures. And that long repeating structure, if you just continue on like this, that is what is known as a core sugar. It's basically monosaccharides, which is just a single sugar molecule like glucose, fructose, galactose, bonded together in this predictable manner like this forever. Now, there's another concept that we need, which is a complex carbohydrate or a complex sugar. And basically, complex sugars are when you mess it up. When instead of just doing the linear pattern, so let's do a bit of the linear pattern, but then you also have other sugars bonded onto the different hydroxyl groups. So if we just draw this one here, basically this sixth carbon up here also has a hydroxyl group, so you could make a similar bond like this. But that messes up the pattern and you get much more complicated sugars, and that's what's known as a complex sugar, where you have basically new sugars linked to the old ones of the carbohydrate chain, but you don't link them in this simple repeating pattern from the first hydroxyl group to the fourth hydroxyl group. Instead, you start linking them off of these other chains, basically, and then you get a complex sugar. So basically, in the endoplasmic reticulum, simple core sugars are added to the structure of CFTR. So CFTR at the moment is just pure protein, it's amino acids. But you're going to, it's a more complicated structure than that. So you're going to add on sugars to modify its structure, basically. And in the endoplasmic reticulum, the type of sugars that you add on are core sugars. So you add on the core sugars. Then what happens is you traffic the CFTR to what's known as the Golgi Apparatus. So here is my best picture of the Golgi Apparatus. So the Golgi is a system of interlinked membrane-bound structures. And basically what's going to happen is that CFTR is bound in the membrane of this endoplasmic reticulum. The endoplasmic reticulum is going to spit off vesicles, effectively, with the CFTR in. And these vesicles, CFTR, and these vesicles containing the CFTR are going to deliver it to the Golgi apparatus. Okay, so once it gets to the Golgi, so let's draw it in the Golgi now. So let's draw CFTR over here. Once it gets into the Golgi, it undergoes further modification. So you start adding complex sugars to it. So in the endoplasmic reticulum, you add core sugars, and in the Golgi, you add complex carbohydrates. So I'll draw I drew a simple sugar there, so I'll draw that same simple sugar. So maybe what happens is the Golgi apparatus starts modifying the core carbohydrate so to turn it into a complex carbohydrate. So let's draw some bits coming off like this, which is where you've attached these new sugars basically onto usually the 6' hydroxyl group. Okay, and then finally what happens is the completed CFTR goes into another vesicle and is trafficked to the apical membrane of the cell. And that's all good. So here's the CFTR going off to the apical membrane of the cell. There is also a process for taking it out of the apical membrane of the cell. So once it does get to the apical membrane of the cell, you need another process for removing it from there, should you decide that there is too much of it. And the process is basically Khafre-mediated endocytosis. So here is our CFTR happily up in the membrane now. And basically another process can remove it and return it back into a vesicle, which is Khafre-mediated endocytosis. And these vesicles can then, these endosomes as they're called then, once you've endocytosed something, it's then called an endosome rather than a vesicle. This endosome can then fuse with a lysosome if you want to destroy the CFTR. So this could go onto a lysosome. Okay, so that's the process by which you traffic CFTR to this apical membrane. So this is Khafre-mediated endocytosis here. So basically what happens in Khafre-mediated endocytosis is that this protein Clathrin binds to the undersurface of the CFTR and forms like a net which starts pinching the membrane downwards, and it eventually sort of covers the entire vesicle and just sort of gradually forms an endosome like so. I shouldn't call it a vesicle. These are endosomes. These are vesicles. These are endosomes. The proteins that are in the membrane of an endosome are different to the proteins that are in a vesicle. So endosomes they really did come from the apical membrane, so they have the same sort of proteins in their surface as the cell membrane has, whereas vesicles have very different proteins in their cell membranes. Well, not the cell membranes, sorry, they're phospholipid bilayers. Okay, so we'll cut this video here, and in the next video what we'll do is we'll discuss how this process is disturbed by a mutation in the CFTR gene, which is very common in cystic fibrosis.